introduce our speaker for today, Dr. Madison Trowbridge. Madison is a spring scientist with the Southwest Florida Water Management District. She holds both a bachelor's and a PhD degree in molecular biology from the University of South Florida. She works with submerged aquatic vegetation and water quality now, but her background is in nutrient cycling and microbial ecology. She's published several papers, but there was one recently that came out in December, analyzing 20 years of SAV data in the Chassahowitzka River system, and that's what she's going to talk to us about today. So thanks for being here, Madison, and I will turn it over to you.
Thank you so much. I'm going to go ahead and share my screen. Can you all see everything okay? You can hear me fine. Wonderful. All right. As mentioned, I am Dr. Madison Trowbridge. I'm the spring scientist at the Southwest Florida Water Management District, and today I will be presenting on indicators of salinization in spring-fed rivers using submerged aquatic vegetation. The info presented today is, as mentioned, from a recently published paper in the Florida Scientist. If you are interested in a copy, you may reach out to me via email, and I'll have my email address on the end, and I will provide a copy to you. I do want to note that while this paper is focused on the Chassahowitzka River, this does apply to all of our coastal springs, which I'll touch up on a little bit later. As the spring scientist, my focus is on springs within our district. Our district spans 16 different counties, from Marion County in the north all the way to Charlotte County in the south. However, most of our springs are in the northern part of the district near the Nature Coast Aquatic Preserve. We have 5 First Manitou springs, and these are the largest springs by the volume of water that they discharge. These springs discharge over 100 cubic feet per second, which is over 64.6 million gallons per day. It is a lot of water. These springs make up in a region which we refer to as the Springs Coast region, and they include Weehee Wachee, Chassahowitzka, Homosassa, Crystal River, Kings Bay, and Rainbow River. 4 of these springs create rivers that eventually flow to the Nature Coast. And although these springs have their own protections as well as their own management plans, it is recognized that these systems do influence the Nature Coast Aquatic Preserve. And this was discussed in the Management Advisory Committee meetings as well as being incorporated into the management plan for the Nature Coast Aquatic Preserve. These springs are internationally known and recognized, and for really good reason. They are beautiful systems to be able to work on. They have clear blue waters, and they're home to many different plants as well as animals. Because they have 72, 74-degree temperatures year-round, these coastal springs provide warm water refuge for manatees in the winter. When we think of springs, we typically think of mermaids and manatees, as well as other components of the freshwater systems. So for example, with Crystal River, it's known as the manatee capital of the world. However, our coastal springs are also influenced by the coast, and as a result, they have both freshwater and saltwater species. So for example, in Weehee Wachee, we have our resident dolphin, which has been nicknamed Chip, that can often be found in the river. He's even made an appearance up at the Head Springs in Buccaneer Bay last year. There are many different stingrays that can be spotted just off the docks of the Homosassa Springs State Park near the headwaters. We've also seen snook as well as bull sharks in the rivers. And I also have a photo on the right which shows the pelicans that will often hang around some of our coastal springs, including at Three Sisters Springs in Kings Bay. Like our seagrasses offshore, submerged aquatic vegetation, which I'll be referring to as SAV, really helps to support our springs habitats. SAV is essentially just benthic underwater vegetation, but it differs from seagrass because these are freshwater species. So seagrasses are halophytes, meaning that they need salt to live, but the freshwater, for the SAV in our springs, these are freshwater species. But like seagrasses, they also provide multiple ecological benefits to our springs. They help to stabilize sediments, they're uptaking nutrients, which helps improve water quality, and they also provide food and habitat for wildlife. There are more than 25 different SAB species found in springs across Florida, but they all can fall into one of three categories. So the first category we have is the desirable or native species, and this includes our Balasinaria americana, which is often referred to as tape grass, but is also locally known as eelgrass. We also have undesirable species, so these are primarily going to be invasive species such as hydrilla. And then we also have algae. So algae is technically native, but it has been considered as undesirable because excess of algae can indicate ecological imbalances within our spring systems. Today we'll be focusing on the SAB in the Chaswitzka River. The Chaswitzka River is located in southern Citrus County, right at the border between Citrus and Hernando County. And if you haven't been to the Chaswitzka River, it is a great place to visit, and I think it's a great homework assignment for everyone if you haven't visited already. Most of this river is surrounded by state land, so it has this really old Florida feel, and I like to describe it almost as like a Jurassic Park, but without the dinosaurs. This river is short, it's shallow, and it's tidally influenced, and you can see the coastal influence actually here on the map. So near the Head Springs area, we have a lot of the green, which is the typical upland wetland species, but then you'll see in this aerial that it transitions to more of this brown color or brown tan color, where the river transitions to a salt marsh. The district has been mapping SAB in the upper portion of the river for over 20 years, and we map at 25 designated locations, which we refer to as transects. Transects are essentially just an invisible line from one bank of the river to the other, and at each of the transects, we deploy 5 quadrats. And a quadrat, which you can see in the photo, is essentially just a square hula hoop that allows us to capture the approximate coverage of the vegetation. We toss out 1 in the middle and then 2 to either side in order to capture the variability from one bank to the other. Sorry about that. All right. The SAV in the Chaswitzka River is very variable. All these photos were taken at one transect near the Head Springs over the course of about a year and a half. At first glance, there's not really any clear patterns with these changes. We'll see these patches of SAV appear, then disappear, they'll move to a different location. In short, the system is very dynamic and it is always changing. So what we wanted to do was we really wanted to investigate whether there were any community patterns within the SAB, and we also wanted to understand the drivers of the SAB community in order to help us make better informed management decisions. So what we did, whoops, sorry, the first step that we wanted to take was looking at what SAB species was there. So we took 20 years of the data pretty much 20 years of our data from what we had presently all the way to when we started mapping SAB. And we looked at the average percent of the species present in order just to see what is the most common species that we're seeing. So let's look first at our rooted species, which are highlighted. The most common that we see are Balasinaria americana, which, as I mentioned earlier, is known as tape grass, but also as eelgrass locally. The Balasinaria is very common within a lot of our springs. It makes the dense beds that you can see in the photo up on the right, and it is a freshwater species with some salt tolerance. Then we also have Xanachellia palustris, which is shown on the bottom photo, and this is a small, very short species. It doesn't create the same kinds of beds that the Vallisneria does. I think it really looks like catgrass, in my personal opinion, but it does tolerate greater ranges of salinity. Now let's talk a little bit about the algae. There are many different species of algae within our springs, and for our SAB mapping purposes, we do not do an in-depth algal identification. Instead, what we do is we categorize algae as filamentous algae, and this will include species like Limbia and Spirogyra, which are more commonly associated with our springs. However, we do see in our coastal springs marine drift algae. Chaetomorpha is the most common of these species, and it often looks like underwater tumbleweeds. So it'll come in and out with the tides. You can see a photo of it here. It's very kind of like hair-like, and it'll just kind of roll along the bottom. Since the mats are generally pretty homogeneous, we do call these out separately when we're doing our monitoring efforts. So as a result, what we're seeing in our mapping efforts is we'll have filamentous algae generally referring to our freshwater algae, and then the marine drift algae are typically categorized separately. So now that we know the key players and for the rooted vegetation and the algae, we wanted to take a deeper look into these spatial patterns. And to do that, we utilized a principal coordinate analysis, which is also referred to as PCO. For those of you that are not familiar with PCO, this is a multivariate technique that we utilize in order to do data visualization. So points that are closer together are going to be more similar, and then vice versa. Points that are farther apart are less similar. Essentially, what we're doing is we're looking for patterns within the data set, and it's kind of like poking the data with a stick, if you will. And the way we do that is essentially just by color coding based on the presence or absence of the SAB that's present. So this PCO is now color coded by SAB type. The light blue is when freshwater algae is present. The red is when the chaetomorpha, the marine drift algae, is present. And the black is when both the filamentous algae and the chaetomorpha are absent. I did want to note that we did look at many different combinations of the SAV within this data set, but this is the one that was the most interesting and what I'll be focusing on today. So as I just mentioned, the PCO is based on patterns. So what we're seeing here is two groupings. We have the freshwater as well as the saltwater algae. And to underscore this, the middle area where neither are present is between these two these two groupings. So what we're seeing is this gradient from the freshwater macroalgae to a marine drift algal community. And this was our first clue that salinity may be driving patterns within the SAB communities in the Chaswitska River. We're now looking at two graphs that compare the percent coverage to the specific conductance, which we utilize as a proxy for salinity. So the x-axis is the specific conductance, essentially salinity, and the y-axis is the percent coverage. On the left, we have the filamentous algae, and the right, we have chaetomorpha. The purple lines are low-S curves in order to show trends in the data, and for comparison, I have put a vertical red dashed line to indicate the known salinity tolerances for the freshwater SAB. So looking at the graph on the left, we're seeing that the filamentous algae coverage tends to be higher with the lower salinities and decreases as you get closer to that salinity threshold. We do see a little bit of a bump here, but when we did some additional analyses, what we found was this was often attributed to when we had both non-homogeneous, so heterogeneous clumps, so filamentous algae mixed with chaetomorpha. Looking at the graph on the right, we see kind of the opposite approaches. We see that closer to that salinity threshold, we have greater chaetomorpha that is present. So what we're seeing is that as salinity is increasing, the percent coverage of the filamentous algae decreases, and the cover of the chaetomorpha is increasing. And these patterns were also confirmed with statistical analyses in R. So shown here are effect plots, which was part of a linear regression model in order to predict interactions between two variables. So let's look at the graph on the left as an example. So the transect number is on the x-axis, and the specific conductance is on the y-axis. The line is essentially a center point of the model, and the shaded gray is expected values. So for example, if you look at transect 2, we can see that the estimated specific conductance range would be approximately between 3,900 to 4,200 microsiemens per centimeter. So what this does is it helps us to look at trends. And so we looked at the trends showing the trends for the transect is showing that increasing specific conductance with downstream distance. But now let's look at the trends for the chaetomorpha as well as the filamentous algae. So the graph in the middle shows the chaetomorpha versus the specific conductance. And what we're seeing is that this graph has a wide range of variability. We did not see any statistical relationship with conductance and the chaetomorpha. So we recognize that there is another variable at play here, and we suspect it may just be because these species are coming and going with the tides. The graph on the right shows the filamentous algae and specific conductance coverages. So what we're seeing now is an inverse relationship between the coverage and specific conductance. So we're seeing greater coverages in the freshwater areas, which just supports what we just saw in the previous slide. And we did find that this was statistically significant. So what we see is that the filamentous algae are limited to the freshwater regions in the river, and the chaetomorpha is coming in almost as an opportunist and is filling in the gap, so to speak, where filamentous algae is absent. From here, we wanted to understand where in this river this is occurring. So we essentially worked backwards. So within the literature and what we just saw, we see that about 3 to 4 parts per thousand, which is again shown by that red line, is about the threshold for the salinity tolerance for freshwater SAB and as well as for freshwater macroalgae. So we wanted to know where in the river do we find these salinities that exceed this threshold. And so we used the graph shown here, which shows the specific conductance, the salinity by the transect, to determine the locations. I also corroborated this with the macroalgae present in order to help identify regions where we would expect to find certain types of macrophytic species. So the first river region is characterized by the tidal freshwater habitat. So the tidal freshwater habitat will have lower salinities, it will have more freshwater macroalgae, so more of that Lyngbia, the Spirogyra, and we're also going to see more of the freshwater macrophytes such as the Balassinaria. So this area is going to be what we would typically think of when we think of a tidally influenced freshwater spring. Next, we have the estuarine zone. So this zone is characterized more by elevated salinities. It'll have more of that marine drift algae like the chaetomorpha, and then it will also have more of those brackish macrophytes, so the rooted vegetation like the Xanachalia. So this area is going to be more of an estuarine community. And then one of the things that we started to notice was that there is this transition zone between the tidal freshwater habitat as well as the estuarine zone. And this transition zone will sometimes be fresh, but it will sometimes be brackish, and it'll have a mixture of both the freshwater and the marine drift algae, as well as a mixture of the freshwater and the brackish SAB. So it'll have both the Balassinaria and the Xanachalia together. What we also see within this area is that it tends to have more community flux than the other two regions. So we took these regions and then we displayed them on the map as shown. And these so this map now shows the approximate locations of these different ecological zones within the river. Now, this model has two primary functions. First, it can help to identify the types of communities that are expected in the different river regions, which can, of course, help drive management actions. And the second, it also provides a baseline to help track how these ecological zones will change over time, such as from factors like climate change and sea level rise. We also found that when we're looking at hydrodynamic modeling alone, it did not necessarily predict the locations of these zones, as the SAB tend to have more of their own ecological niche over compared to what the say a salinity model would show. In some of the initial Springs SAB literature, it was suggested that, several decades in past, there was this suggestion that SAB and Springs are very stable. But coastal springs are actually quite dynamic, and a lot of this appears to be driven in part by the salinity fluctuations that we can see within these systems. But we can use this information, of course, to our advantage. So we know that salinity is a known driver of SAB in our coastal spring. So looking at the presence or absence of SAB species can help us to determine the locations of the different zones within the river. And the understanding of these locations, of these regions, may impact the approaches to different management actions within our coastal systems. So, for example, macroalgae like Spirogyra and Lingbia may interact differently with nutrients from our spring vents than marine drift algae will. So, for us at the district, this will also allow us to track and potentially predict the changes within the coastal systems due to those factors, like I mentioned, the climate change and the sea level rise. However, for other folks that are working with different systems that may not have an abundance of rooted macrophytes, so those rooted vegetation, macroalgae can be used instead in order to help identify these regions. So in short, the presented model has a variety of uses for many different aspects for coastal springs management, and perhaps even for other coastal systems. This schema really highlights the findings of this work, and it really helps to provide a great visual summary. So coastal springs are unique, and then they have both the freshwater and the coastal, or the freshwater areas and this more saline areas, and that they have this coastal influence with them. So as a result, we see that these systems are very dynamic and may have different zones between the head spring and the mouth of the river, which may or may not change over time. So this shows that not only do our coastal springs help to shape the nature coast, but that the nature coast is also helping to shape our springs. So with that, I thank you all for your time and attention. And I know I'm a little early, but I can go ahead and take any questions if there are any. And feel free to scan the QR code to sign up for our newsletter. Thank you.
Thank you so much, Madison. That was great. I'm seeing lots of clapping in clapping hands too. Does anyone have any questions? If you do, feel free to type them into the chat. You can also unmute yourself and ask the question directly as well. And while people are thinking of their questions, I can get started. You're sort of hinting a little bit about that this transition zone might move a little bit. And what are the implications of that? Is it more sedimentation? Like what are the implications of having more that transition move upriver?
So with that transition zone, we're able to, for all the zones really, we're able to see both the short and the long-term impacts over time. So obviously long-term impacts are going to include like sea level rise, the climate change, where we can see if it does encroach, we're seeing more salinity within our spring systems. That can be used to see what's going on over time within our springs. However, there's also some short-term impacts. So as I think many of us may know that we saw impacts from Hurricane Idalia and essentially pushed a lot of salt water up into our coastal spring systems. So because there was a lot of salt water, it did affect the SAB communities. And as we're continuing to monitor the impacts, we're able to understand, are these zones changing? Are they moving? Are they reestablishing the same ways? And we can understand those zones because if there's a management action, then we can kind of focus on those specific areas. So for example, if we're in the estuarine zone, or we know we're in the estuarine zone, maybe oyster restoration would be more appropriate for that area versus, let's say, SAB planting as an example.
Great. Jim has a question. You're welcome to unmute yourselves and ask the question.
Thanks. Yeah, typing it would have been hard on this phone. But anyway, I appreciate hearing this stuff and going back to your decades old thing that is pretty stable. and having studied the macroalgae in that area back then, have you been able to look back close enough to those studies, including the ones done for the Cross Florida Barge Canal that were extensive on the macroalgae throughout that region? Can you relate that to the fact of the instability or the dropped level of water coming from these springs?
So to answer your first question, the SAV data that we have for the Chaswitzka River system is based on the district's data. So we began collecting 20 years ago, and that's what was presented on today. We don't necessarily have some of those like detailed macroalgal IDs, unfortunately, just due to the nature of our study. As far as your second question regarding, I believe it was was it salinity or can you remind me the second question?
Pretty much, where you're saying decades ago, the stability of the springs or that the macroalgae within the spring system were, pretty much consistent, and they were, from my observations way back then, also. And I'm wondering if there is anything that can relate that directly to the flow volume from those springs, which actually maintain more of a standard pattern than the extremes it goes to today.
Oh, I understand your question a little bit better now. So when I was referring to the decades ago, for Chaswitzka, we actually don't have some of those historic references, unfortunately. But some of the initial springs work that was conducted in the 1950s, not on Chaswitzka, but in springs in general, those were suggesting that springs typically didn't change as much as we thought, or as they thought back then. Now, since then, there's been a lot more work to suggest that there is a lot more variability than what was originally done. We can look at changes over time, especially with some of these modeling, to kind of understand some of those patterns. And that's what this this kind of model that was generated from this paper is trying to help understand is, at least right now, especially, we have that snapshot in time. And so we can use this as a way to understand future impacts. Does that help kind of clarify your question?
Unmute. Yeah, you're kind of in the ballpark anyway.
Okay.
It's kind of fun to see, and definitely there have been changes to that whole
Yeah.
system, but terrestrial and not. So appreciate your research. Thanks.
Thank you.
There's a question that you kind of alluded to already, but you might have some more to say about it. Eugene Kelly asked, is there historical data that confirms the dynamicism that you see now, or might more recent changes in spring flow be playing a role?
That's a great question. So in the Chaswitzka River, we don't have that information, just because there wasn't a lot of historic data collected on SAV. However, in some of our other coastal systems, we do have some evidence. It's not the same extent of impacts of salinity, but we do see this in some of our neighboring springs, such as Homosassa, where we do see shifts in the SAV community, meaning the type of SAV that's present. We will see there was some work done in the 1950s where you could see that the Valles scenario percent coverage was decreasing as you moved downriver. It seems that in more recent years, at least especially within the last decade, there's more and more research into the salinity aspect, which I don't think was as focused on, previously. But we do see that there are some historic, at least knowledge that there were changes due to salinities even back then.
Yeah, and there's a follow-up to that one. Impacts associated with nitrification are also seen in most of these spring systems. Could increases in nitrogen play a role that confounds your interpretation, or do you have a basis for rejecting any role of nutrients in SAV distribution or zonation?
So we don't discount the role of nutrients. We do recognize that nutrients may play a role. It's more from what we've seen. I did look into some of the nutrients within the Chaswitzka River, which is generally about the same throughout, at least in the area that we were working on. But one of the things that I think would be really interesting to look at would be how nutrients may impact one region versus another. So for example, how is the freshwater SAV, the tidal freshwater habitat being impacted by nutrients versus the more estuarine portions? Are they reacting the same way? And I think currently we may, um, there's kind of some assumptions that they're reacting the same way, but there could be some areas that we see more of an exacerbated response than others. But that's something I'd love to look at kind of moving forward as a next step.
I have another question.
Sure.
You did a lot with some really coarse data about what species are there. When you're
Mhm.
working with these data, are there things you're like, man, going forward, we should really change our monitoring program and look at more specific species, or is this, you know, the best approach for now?
So we feel that, I mean, we're always trying to improve, and I'd say when we first started, we definitely had some improvements from what we did 20 years ago, but I feel like what we have now is really good to understanding just how these systems are changing. And we have a lot of different tools. I'm just showing you one of these tools, which is that SAB monitoring, but we also have our continuous monitoring network at the district for water quality, which looks from the head springs all the way out to the Gulf and to the Nature Coast Aquatic Preserve. We have flow data. We have a lot out there, and we're able to utilize many different tools in order to be able to understand how these systems are changing. And the SAB mapping, it really does help to understand some of these short and long-term changes as well.
Any other questions from the group? All right. Well, thank you so much, Madison, for your presentation. Thanks everyone for joining. Hope to see you next month when we'll hear from Bea Diaz about seagrasses and hurricane impacts. Thanks again.
Thank you.